Physiology of the central nervous system. Nervous system may be divided into the central nervous system and the peripheral nervous system. Central nervous system consists of the brain and the spinal cord. Peripheral nervous system consists of different nerves and ganglions. Ganglions are the collections of bodies of neurons outside the central nervous system. On the other hand, functionally, nervous system may be divided into the somatic nervous system and autonomic nervous system. Somatic nervous system controls striated muscles. Autonomic nervous system controls endocrine glands, internal organs, and is unconscious. The basic mechanism of the nervous system functioning is a reflex arch. We will draw the reflex arch of the somatic nervous system. Before, we should define the reflex. The reflex is a response of an organism to the stimulation with participants of the nervous system. The reflex arch begins with the stimulation that affects the receptors, then afferent neuron, or sensitive neuron, which goes directly into the spinal cord. This is the spinal cord. We have gray matter. Here are the bodies of neurons, and the peripheral part, white matter with myelinated and unmyelinated nerve fibers. So the afferent neuron enters the posterior horns of the gray matter. Here the body of interconnecting neuron is located. It produces an axon and it produces the synapse on the body of the efferent neuron, which is located in the anterior horn of the gray matter. And efferent neuron, here is the efferent neuron, produces an axon that is leaving the spinal cord, and in the anterior roots of the spinal cord it reaches in the somatic nervous system the skeletal muscle or effector. Here we have effector. The body of the afferent neuron is located in the spinal ganglion. Spinal ganglion that is in the dorsal roots of the spinal cord. This neuron is pseudo-unipolar, so it has the body, then single process is produced, and then this process is divided into the axon and dendrite. So it seems to be a one single process. It is not so. That's why it is called pseudo-unipolar. Nerve centers. Nerve centers are the collections of neurons which fulfill a certain function. For example, there is center of respiration. This center is including the neurons in the medulla oblongata, which coordinate the respiration. Or there is center of hunger, which is located in the lateral hypothalamus, and the activation of these neurons initiates the motivation of hunger, etc. In fact, we have nerve centers in narrow and wide sense. In narrow sense, this is the localized number of neurons, which is crucial for the certain function. If we will destroy these neurons, the function will disappear. Again, center of respiration in narrow sense is located in the medulla oblongata. In wide sense, the nerve center may also include the neurons which are located maybe at relatively long distance from the nerve center in the narrow sense. Example, in the respiration center in the wide sense, we also include the brain cortex, which makes possible conscious control of respiration, limbic system that adds an emotional impact of emotions upon the respiration. We also may include other parts of the brain which are not crucial. If they will be eliminated, then respiration will not stop. But these structures, they help to adapt and to make the respiration better. Now we should repeat the properties of the nerve centers. We have a group of properties of the nerve centers which are explained by the properties of the synapses, which binds the neurons in the nerve centers together. From here we may say that there is one-way conduction, high fatigability, central delay, which is a sum of synaptic delays in this nerve center, sensitivity to chemicals and to oxygen deficit, low lability and other properties of synapses. Other properties of the nerve centers are specific. For example, we have plasticity of nerve centers. Plasticity means that nerve centers may to some extent learn and change their functions. For example, after the stroke, the patients may recover quite quickly sometimes because the neurons which were not damaged, which were not killed by the stroke, may learn and substitute the eliminated neurons. So plasticity, dominant principle. Dominance principle means that at each moment some center of the brain, some nerve center becomes the chef, becomes the most important, and it suppresses the activity of other nerve centers which coordinate less important functions. And an example, if a person is hungry and is thirsty at the moment, then thirst will be dominating because it is most important for survival, more important than hunger. And after the person will satisfy the thirst, then he will feel the hunger. Next property is the tone of the nerve centers. The tone means some extent of excitation that appears even if the center is inactive. The tone of nerve centers is explained by the humoral regulation factors, by hormones, for example, level in the blood, and on the other hand, by the afferent inputs in that nerve center. For example, the tone of respiration center in the medulla oblongata depends on the content of oxygen in the blood and carbon dioxide in the blood, and on the other hand, by nerve impulses from the receptors in the lung, for example. Other properties of the nerve centers will be explained by a certain relationship between the separate neurons in the nerve center. Now we will speak about the impulse conduction in the neuronal nets, and we will illustrate the typical situations that will explain how the neurons interact with each other. The first property in that group is called convergence. Convergence means that to the body of one neuron, this is the body, this is an axon and the nucleus, we may simultaneously observe the convergence of several other excitations of other two or more neurons. So the excitations converge on the body of the one neuron. Next property is called divergence. We also see one neuron, an axon, and an axon may have a branch. So this is a branch or collateral. And this axon and its branch may go to different neurons in the nervous system. So we, in that case, observe the divergence of excitation in the central nervous system. Another type is called reverberation. In that case, we see several neurons. Second neurons may produce the collateral, and then we may see the loop. Several neurons, and excitation may go back. So, if action potential is produced in that system, it will circulate in this neuronal gap or neuronal loop for a while, and then may go away. This type of neuronal interaction may explain the short-term memory. The information may be kept in the brain, in mind, in the course of this circulation, but it is not long-term because it will not circulate for a long time. Another type is called multiplication. Here we have a neuron that conducts the impulse to another neuron. So if action potential, we will draw an action potential, is produced, then it may go straight and may go to collateral. And here we will have another neuron. Also we have another neuron like that, and another one more neuron. So what will happen in that case? Synaptic delays will be added, and to the second neuron from the first we will observe not one action potential but three action potentials because of these synaptic delays, and that is multiplication really. We had one spike and now we have three spikes. Multiplication. Next question is about the function of a neuron. So neuron may have different functions, but the main function is an integrative function of a neuron. We should also repeat the convergence. Again, we will speak about the convergence. So convergence may be divided into the multibiological, when one neuron conducts nerve impulses which may code the information, for example, about the hunger. And here we may see the neuron that coding the information about thirst, for example. And the information is coded by the neuronal pattern, which is in fact the consequence of spikes, which go in the intricate way, which may have different interspike intervals, and the neuron that is in the center that receives this information is not only changes the potential of the membrane, but also through metabotropic receptors it changes the activity of enzymes and genetic factors in the cytoplasm. So the neuron produces a quite original pattern that will not be similar to any of inputs that this neuron receives. This is called integrative function of a neuron. By the way, the convergence may be not only multi-biological, as in that case, hunger and thirst. It may be multisensory. For example, here we have light, and here we have noise. Multisensory or sensor biological convergence, when one neuron is coding some sensory input, for example light, and another biological information, for example hunger. This is sensory biological convergence. Now we will speak about the methods of clinical investigation and scientific investigation of the central nervous system functions. One method is called electroencephalography. This is called also EEG. The graph is called electroencephalogram. Electroencephalography is the method that registrate the sum of all electrical activities of the brain. So it summarizes the electrical activity of the brain and represents this activity in a graph. The graph looks like that, electroencephalogram. So it may have different rhythms or different waves which are characterized by different frequencies and amplitudes. The waves with lowest frequency from 1 to 4 Hertz are called delta rhythm. Delta rhythm is characterized also by the highest amplitude, up to 200 microvolts. Next rhythm is theta rhythm. From 4 to 8 Hertz. And the amplitude is lower, up to 100 microvolts. By the way, the significance, functional significance, delta rhythm is the rhythm of a deep sleep, when the phase also is called delta sleep, delta phase of the sleep. Theta rhythm is the rhythm of emotional stress. The person is under the stress. Next rhythm is alpha rhythm, from 8 to 12 Hertz. Alpha rhythm is lower amplitude. about 70 microvolts, and about 30 microvolts is beta rhythm, which is from 12 to 35 Hertz. Alpha rhythm is observed when person is in resting situation with closed eyes. With closed eyes, the alpha rhythm is most prominent, and the beta rhythm occurs when person is solving mathematical or any other tasks, is the intellectual activity. The investigation of EEG is used in the clinical practice for diagnostic of epilepsy. Seizures are reflected very well in the electrical activity of the brain, and to promote, to activate this special type of activity, photostimulation is widely used when the flash activates the seizures, and hyperventilation probe when person respires with high frequency, and the deep respiration is used. Another method is called microelectrode method. So microelectrodes may be with very thin tip, which may be close to the neuron. This is a neuron, and we can use an amplifier to amplify the electrical activity of a neuron. So with this method we can use the amplifier to see an action potential, for example. And we can also use microiontophoresis to inject by electrical current different molecules of different transmitters and drugs to perineuronal space to investigate how the electrical activity of the neuron will respond to different types of humoral factors, for example, of different hormones or peptides and so on.